Hello again, biologists. Mr. Kinnear here with another video lesson for higher biology. We're on Unit 1, DNA and the Genome, and today we're going to look at gene expression, which is key area 1.3. So we're moving on in our journey of understanding DNA. We've looked at the structure and organisation in key area 1.1. 1.2 was talking about replication of DNA, and we're now on to gene expression, which is about making proteins from the DNA code. That will then move on to differentiation and the structure of the entire genome, understanding mutations, evolution, and genomics. So the key concepts we're going to look at today: what is gene expression? How is that DNA code converted into a primary transcript? How is that transcript modified to produce a mature transcript? And what happens at the ribosome to produce a polypeptide? So those are all the steps involved in gene expression. Gene expression ultimately is about the production of proteins based on the code that we have stored in our DNA. You and I look different because we produce different proteins determined by small differences in our genetic code, which determine things like our hair colour and eye colour and other bits and pieces. We also share a huge amount of our DNA producing identical proteins in our cells that are involved in cellular processes. But what we would say is that the DNA that's in your chromosomes is the code or recipe book to make you who you are. And as we talked about in National 5, there's a couple of steps between DNA and protein. We didn't call them these names: transcription, which ultimately is about the recipe being copied from recipe book, and translation, which is about taking that recipe and converting it into the final product. So that's what we're going to be looking at in part 1 and part 2 of our video lesson today. Part 1 being transcription, part 2 being translation. Gene expression is controlled by the regulation of transcription and translation, the two processes that I've just mentioned. But what does that mean? It means that anything that makes transcription happen and or translation happen promotes the production of a protein. Or anything that can stop transcription happening or translation happening will ultimately stop the production of a protein. So those are things that we're going to look at later on, but you need to know now that if we managed these two processes, then we manage the production of proteins. So we'll start the process by looking at transcription, and as you can see from the Amoeba Sisters cartoon, we're going to look at mRNA. Now that's something we talked about in National 5, but obviously again we'll add some more detail to it this year. In this process we're making a transcript, and a transcript can be referred to as a different format of something. So for instance, someone makes a speech in Parliament, that speech is then typed up and can be read instead of listened to. And that's what we're doing with DNA. We're converting DNA into RNA. So it's not an exact copy, it's a complementary copy. We're not doing DNA replication, we're doing transcription. The sequence of bases that's put together on our primary transcript is determined by the complementary base pairings. And we talked about that in the last lesson when we reviewed the structure and function of RNA. And that's put together by an enzyme, RNA Polymerase. So now we've talked about a second type of polymerase. I now need you to be specific when you're talking about polymerase and mention RNA polymerase or DNA polymerase in the right places. Here we have a gene ready for transcription. Upstream of that we have a promoter, an area of DNA which tells the RNA polymerase where to bind. Downstream of that, and it's thousands of bases downstream, at the end of our gene we have a terminator area. So a sequence of DNA that tells RNA polymerase to stop. RNA polymerase binds to the promoter region, and it does two jobs in one go. It breaks the hydrogen bond, so splitting our double helix, and also adds the free RNA nucleotides in a complementary sequence, creating our primary transcript as it moves along the gene. And as I said, as it reaches the terminator sequence, RNA polymerase is released. And that's our primary transcript, our complementary copy of our DNA sequence, complete. But it's not over yet. We've produced a primary transcript, which is a complementary copy of what's on our DNA strand. However, we're going to cut out some bits bits that are not useful to the production of this protein, and that's called splicing. That will produce a mature transcript which can leave the nucleus. We're going to modify this primary transcript. So what we're saying is that the gene contains areas which are not useful to the production of this protein, the introns. And because our primary transcript is a complementary copy, it also contains introns. What we want to do is cut out the introns in splicing and stick together the exons to produce a mature transcript which can go off for translation. So it's our introns of the primary transcript which are non-coding and are removed during RNA splicing. The exons are coding regions and are joined together to produce a mature transcript. And that mature transcript is what passes out of the nucleus into the cytoplasm so that translation can take place. But if that's a bit complicated, an easy way to remember it is introns interrupt the code, exons are expressed. So that's an easier way to remember that bit of information about introns and exons. Last year in National 5, we gave you the definition of a gene as a sequence of DNA that codes for one protein. And for many years we thought that to be true. But developments like the Human Genome Project have shown us we contain 20 to 25,000 genes, but over 1 million proteins. So that means that something's happening to multiply the amount of protein that we can get from one single gene. One of the steps we can take to make different proteins from one gene is alternative splicing. And that's about editing in different ways. So putting different sequence of exons together for translation. So in this example, we've got 4 exons on the gene, and we alternative splice to create 2 different types of mRNA transcripts. And I'm sure there's more than 2 different sequences you can have there. So many different mature transcripts of mRNA can be derived from one section of DNA, which go off then to produce different proteins. So that's us gone through the first process within gene expression, transcription. So we've taken our genetic code stored in our DNA and produced a mature mRNA transcript. Now we need to translate that from mRNA transcript into a polypeptide or a protein. Earlier in this video I gave you a definition of the word transcript. Translation is a word you'll be familiar with, and it's probably something you've already done in your modern language lessons. You will have translated English into French or Spanish and vice versa. So translation in what we're looking at just now is the conversion of the language of bases into a language of amino acids to make polypeptide chains or proteins, and that's happening at the ribosome. So we're translating that message on mRNA into a polypeptide chain. We know from National 5 biology that ribosomes are where proteins are synthesized. We also know that they are found in the cytoplasm. A new bit of information there is that they are also found on rough endoplasmic reticulum. And again, we've learned this year that they consist of proteins and ribosomal RNA. And ribosomes, again, new knowledge, they can bind 2 mRNA codons at one time. So as you can see here, we've got a chain of mRNA that is feeding into a ribosome. And at the moment, we've got 2 codons there sitting within the ribosome. I introduced you to this structure a couple of lessons ago, the transfer RNA molecule. There's 2 bits that we're interested in. This anticodon here, which is complementary to a codon. And up here, we have an amino acid. And again, each transfer RNA molecule is specific to one amino acid. And I think about it like a taxi. It's one job. Transfer RNA, it's one job. It's a taxi one particular amino acid from the cytoplasm to the ribosome. And we'll talk about why it does that in a second. So our first codon, our start codon, is AUG. That's what initiates translation. So every mature mRNA transcript is going to start with AUG. And the tRNA molecule with complementary anticodon UAC binds to the ribosome. So here we have it, UAC, complementary to AUG. And actually our first amino acid is always going to be methionine. So every single protein strand starts with methionine. The next step is for a second tRNA molecule to come along. Obviously, its anticodon is going to be complementary to the second codon, and then binds the ribosome. And as you can see, our second codon is GGU. So what should be the anticodon on this tRNA molecule? Again, it's mRNA, it's complementary, so we should be seeing CCA. And in this instance, this transfer RNA molecule is going to bring on glycine. Now we have our first two amino acids in the sequence. We need to join them. So a peptide bond, so that again, National 5 knowledge, peptide bond between amino acids to form polypeptides. In this instance, because it's two, we now have a dipeptide. So our first two amino acids in the sequence are joined together to form a dipeptide. This process then just continues. So the first tRNA molecule is free to leave the ribosome. That obviously brings in a brand new codon, a brand new tRNA molecule with an anticodon and a new amino acid. So now we have methionine, glycine, and glutamine. We're then going to form a peptide bond between those two, and then again it just continues until we reach a stop codon. So the stop codon, obviously again being a message to say to the ribosome, that's it, stop translating at this stage, and the polypeptide will be finished. But we're looking at thousands of amino acids within a polypeptide. We are translating mRNA codons to amino acids. Now, if you're to translate English into French in modern languages, you would hopefully have an English to French dictionary to help you out. Likewise, we have an mRNA codon to amino acid chart. So it's less than a dictionary because we only have 20 amino acids to code for. We have a lot more codes than that, however. So a lot of the amino acids have more than one code. So to give you an example, we're going to start off with this codon CAA. And how do we read this chart? Well, we have to look for the first base. So in this instance, we're going down to C as our first base. So we've got the second row. And we go along the top for the second base. So we're going to go along to the third box for CA. And as you can see, the third base, we go down again. So the third base, the third line here, CAA. And as you can see, that codes for glutamine. And you can also spot that glutamine is also coded for by CAG. So let's look at another example. We've got GCU as our second codon. So in this instance, our first base, we have to go all the way down to the bottom for G. Again, we're going to go along, and our second base is found in the second column, so GC. And again, we've got U here on the first rung. So GCU codes for alanine. And as you can see, all of these codes, GCU, GCC, GCA, and GCG, all code for the amino acid alanine. Amongst that, we also have, as we said earlier, AUG for the start codon. It's methionine, so every single amino acid starts with the amino acid methionine. And we also have stop codons. So we have UAG, UAA, and UGA are all stop codons. So that tells the ribosome to stop translating, and that will end our polypeptide chain. So where does all of this happen? This is something you need to know as well. So the formation of a primary transcript of mRNA, you should remember from the last lesson, that's in the nucleus. The modification of that primary transcript to create our mature mRNA transcript, again, that happens within the nucleus. Collection of amino acids by tRNA, so those are scattered about the cytoplasm. So tRNA takes them from the cytoplasm, and the formation of codon-anticodon links, again, that happens in the ribosome. So that's something to be very clear at. So mRNA, its formation and its modification happen in the nucleus. The amino acids are in the cytoplasm, they're collected by the tRNA, and those codon-anticodon links and the polypeptide links, they are happening in the ribosome. So we have created a primary structure. All we've done today is create a chain of amino acids. We start with methionine,
So that's our start codon, and right at the end we have our stop codon. And that effectively, as you can see here, just creates a chain of amino acids, a polypeptide chain. You can also see we've got secondary, tertiary, and quaternary structure, and we're going to look at those in a bit more detail later on. But all we've got today is a primary structure, just a length of amino acids joined together with peptide bonds.
So that's us for today's video lesson on gene expression. Today we've looked at the stages and molecules involved in the process of transcription, which is creating an mRNA transcript from our DNA code. We've also then looked at alternative RNA splicing, which means that one gene can code for many proteins. And then we've also looked at the stages and molecules involved in the process of translation. So that's converting our mRNA transcript into a primary polypeptide structure. And we will look at the further structures in later video lessons. So as always, if you have any problems, questions or queries, get in touch and I'll speak to you again soon. Bye.